Hello and welcome to Oxford Nanopore Technology's Flow Cell Loading Masterclass. My name is Valdemar. I'm a technical application scientist here at Oxford Nanopore, and today I will present and demonstrate flow cell loading, as well as we will have a section of Follow Me Along to load the flow cells from our demo packs that you would have received for this London Calling 2025 conference. A quick reminder, this masterclass is part of a more extended series of masterclasses and lessons where me and my colleagues take you the full path from sample extraction and preparation to sequencing all the way down to analysis of your data generated with Oxford Nanopore Technologies. Additionally, we have a few masterclasses featuring end-to-end applications that are possible with Oxford Nanopore sequencing and technologies. In this masterclass, we'll focus specifically on flow cell loading. A quick note, within this masterclass, we will also briefly touch upon the flexibility and scalability of Oxford Nanopore Technologies, as well as present different flow cells and devices that they can be paired with, and the range of different devices that are available from Oxford Nanopore. Then, we will demonstrate the flow cell loading, as well as do a follow me along session with the demo packs delivered for this conference. So let's move along into what types of flow cells exist and what kinds of flow cells can be used with Oxford Nanopore devices. So Oxford Nanopore technology uses small, tiny nanopore channels to sequence DNA by passing DNA through them. Between the flow cells, the chemistry, the nanopores, and the library preparation is exactly the same. There is no difference in accuracy or in chemistry between the flow cells. The main difference between the two flow cell form factors that we have is the size of the nanopore array and the total amount of nanopore channels. Depending on how many nanopores the flow cell has, it will change how much data can be generated from a particular flow cell. There are two main form factors of flow cells available: MinION form factor and PromethION form factor. MinION flow cells are smaller flow cells that have up to 512 nanopore channels on them and can generate about 25 gigabases of sequencing data. Those flow cells are mostly suitable for smaller genome applications, such as multiplexing multiple small genomes on a single flow cell, low-pass sequencing of large genomes, or targeted sequencing of the same said large genomes or amplicon panels. PromethION flow cell type is our highest capacity, highest output flow cells that are suitable for sequencing and working with really large genomes. With PromethION flow cells, you can generate hundreds of gigabases of data, and you can sequence large genomes in applications such as one human genome per one PromethION flow cell. They have a lot more nanopores, hence are able to generate a lot more data. Yet, between these two flow cell types, the chemistry and the technology is the same. The core part of the technology remains equivalent. Depending on the flow cell type and the flow cell type that's suitable for your application, it will govern what sequencing device you can choose. For MinION form factor, for MinION flow cells, devices that are suitable for it are MinION and GridION devices. For PromethION form factor, the PromethION line of devices is what you should choose, and devices such as PromethION P2 Solo that can be plugged into your computer or into the GridION, PromethION 2 Integrated, which is the device we will be demonstrating loading today, as well as the biggest PromethION 24 device that can accept up to 24 flow cells with 24 independent positions for PromethION. Having reviewed the devices really quickly, I think now we can proceed and focus a little bit more on the flow cells themselves. So before proceeding on loading the flow cell, and the first step today, I will just demonstrate the loading as you would do it in the lab. Quick note on health and safety. When handling flow cells or reagents from Oxford Nanopore, please remember that this is lab equipment, so please wear safety glasses and neutral gloves when you're doing so, as well as don't handle reagents or flow cells in presence of food or in the food preparation areas. After we have used all the materials today, we will ask you to please put all of those materials back into pouch that the demo packs have come in, so that we can dispose of them, so that you can dispose of them in accordance with local regulations. Okay, now that I've said this, I will go on and follow my own advice, put on my safety specs and gloves before we can proceed with loading the PromethION flow cell. Now, I will run through the PromethION flow cell loading first, and then after this section, we will actually do them with the demo packs that you have as part of this London Calling 2025 conference. Now, gloves are on. We can actually take a look at the flow cells themselves. Before we proceed with the flow cell loading, I believe there are a few parts of the flow cell that are useful to know about and pay attention to. So I will open the valve that's located on the flow cell by rotating it 90 degrees. And a few parts that are useful to note. The main part of the flow cell, where all the sequencing nanopores are, where all the channels are, is the flow cell array that is visible on the flow cell through these two rectangular windows. It is located actually slightly deeper below the flow cell. This is the main part and the most sensitive part of the flow cell, so we do not want to interact with it directly, and it is actually inaccessible to the user to pipette things onto it directly. To interact with this sequencing array, there are three main ports on the flow cell through which user can interact with it. The main channel through which we will be priming the flow cell and loading our DNA sample is the priming port that is located under the valve that I've just rotated. This is the main port through which we will interact with the array, load our sample. And if you're only using the flow cell once, if you're only loading the library once, this is the only port you need to interact with. The ports numbered 2 and 3 over here are the waste ports that normally you only need to use if you're performing a flow cell wash for repeated use or if you're reloading the library onto the flow cell. Today, since we're only loading once, we will not actually need to interact with them. Now, I think we can proceed with loading. The priming port that is exposed right now by opening the valve, as you can see, this part of the channel is colored in gray. The channels that are colored in gray have air in them. The channels that are of black color have liquid in them. To begin with, before we can do anything with the flow cell, we need to remove this small air bubble that's located in the priming port. This is because air doesn't pass the current really well, and moreover, the air bubbles on the sequencing array can irreversibly damage the nanopores themselves, preventing them from sequencing. So by any means, we always want to make sure there is no air going onto the array. As a first step, what I will do is I will set up my P1000 pipette to 200 microliters. I will insert it into the flow cell, make sure it's nice and snug there, it makes a seal, and I will very carefully rotate the flow cell dial anti-clockwise to aspirate in a controlled manner, to aspirate a small amount of liquid out of the flow cell until I can see that there is no more air in the priming port. Now I see that small amount of storage buffer, the green colored liquid, came into the pipette tip, so I know that there is no more air, and now I have access to the liquid. I will expel that onto a clean tissue and dispose of it, and the flow cell is ready for priming. Priming of a PromethION flow cell happens in two simple steps, 2 times 500 microliters, which I will do now. So I will set my pipette to 500 microliters and will do the first priming step. I will use my priming mix, which normally would be flow cell flush and flow cell tether mixed together as described in the library preparation protocol. I take the first 500 microliters, make sure there is no air in the pipette tip, insert it into the priming port, and carefully and gradually expel the liquid into the flow cell. This liquid right now, this priming buffer, is going directly onto the array. I will leave a little bit of liquid inside to make sure I'm not introducing any air into the flow cell, disconnect the flow cell, and this way I know that there was no air that went onto the array, so my nanopores will not get damaged. At this point, I will wait for 5 minutes, but because this is just a demonstration and the flow cell is not actually suitable for sequencing, we will do some hand magic and skip the 5 minutes and proceed directly into the second loading step. I don't need to change anything on my pipette. It's already set up to 500 microliters. So I will just finish and do the second step of PromethION Flow Cell priming by adding another 500 microliters of priming buffer into the flow cell, like so. Once again, I always recommend to leave a small amount of liquid in the pipette tip to ensure that you're not accidentally introducing the air. I'd rather not have irreversible damage on the flow cell than risk it when I'm expelling all the liquid. At this stage, once we've done 2 steps, once we've put in a total of 1 milliliter of buffer through the flow cell is ready for loading the DNA sample. And today, as my DNA sample for this demonstration, I will use the library loading beads, which is what the beads that normally would go into the library. And to load the DNA sample, the DNA library, onto the PromethION Flow Cell, I will use the same P1000 pipette, because this pipette is ideally suited for the priming port. It creates the best seal. But this time I will set it to 200 microliters, which is the amount of library that we load onto PromethION Flow Cells. And take my library out of the tube. Check that there is no air at the end of the pipette tip. It is also always recommended to hold the pipette tip vertically to avoid any air coming in. Insert the pipette tip into the flow cell and once again, carefully and gradually expel the library into the flow cell. You can see that the channel is now, the library loading beads are now visible in the channel. So this is where the liquid is going into the array. And as previously, leaving a little bit of liquid behind to avoid any bubbles. At this stage, the library is loaded onto the flow cell and the flow cell is ready for sequencing. So I will close up my tubes. I will close the priming port. This will prevent any evaporation over the 72-hour sequencing runs, so we don't change any osmotics on the flow cell. Pop on the flow cell light shield, like so, to cover the flow cell from the light. Close the cover, and the flow cell is ready for sequencing. Now, we've done with the PromethION Flow Cell, and I will get ready to do the demonstration and follow along with me with the flow cell demo packs. For now, I have recorded a MinION Flow Cell loading video, so let's roll that while I get ready. In order to prime a MinION Flow Cell, first, open the priming port cover. You will see that this area here is of a lighter color than the rest of the channel. This is because of an air pocket that needs to be removed before the flush. To remove this air pocket, use a P1000 pipette to withdraw a few microliters of buffer by either raising the plunger gently or twisting the plunger like so. As you draw back the buffer, you'll notice the buffer front move like this. It is important that you do not draw back too much at this stage, or you'll risk exposing the nanopore array to air via the other end like so. Once we have removed the air pocket, we load 800 microliters of priming mix through the priming port using a P1000 pipette. Another tip to avoid introducing air bubbles into the system is to not expel all of the liquid into the port, leaving some behind in the pipette tip. After the flush, we wait 5 minutes and then open the spot-on tab. Next, we load 200 microliters of priming flush through the priming port whilst keeping the spot-on port open. As we do this, we force the buffer front in the spot-on port to rise slightly and displace any air bubbles that may be there. This again reduces the risk of air bubbles being pushed onto the array during the sample loading. The library needs to be loaded onto the flow cell immediately after the second flush. When you're ready to load the library, use a P200 pipette to add the library dropwise onto the spot-on port. As you do this, ensure the tip only hovers above the port and that it does not come into contact with the port itself. This is because the spot-on port sits directly above the array and we do not want to damage the array or introduce any air bubbles into the system. After you've loaded the library prep and are ready to run your experiment, close both the spot-on tab and the priming valve to prevent evaporation and subsequent change in osmotics during the run. Put on the light shield and close the lid. You are now ready to start your sequencing run. Welcome back. Hope the MinION Flow Cell loading video was nice and clear, and I think now we can proceed to the final part of this masterclass, the one that you've been all waiting for, where we take our flow cell demo packs and load the flow cell together. Now, before we do that, a quick reminder about health and safety. So, as previously mentioned, please make sure to wear your gloves and safety goggles, as well as for this loading demo, we would recommend to have a small amount of tissue ready on the side to expel the excess liquids of the reagents that we will use. Please make sure you're also away from any food preparation or food consumption areas. I will now give you a few seconds to get all of that ready and put your PPE on. And while you do that, I will open up the flow cell demo pack to check what is inside of it. So, as we open the flow cell loading demo pack, we can see that there are a few things inside. I'll just put everything out on the table here and set the pouch aside. What we have inside of the pack is our flow cell, the star of the show today, the syringe with a pipette tip on, which we will use instead of our pipette, as well as two tubes, one with a blue cap labeled FB, flush buffer, which we will use as our flow cell priming mix, as well as another tube labeled LIB with pink cap that we will use as our library. This contains the library loading beads. Now, before we go ahead and start the PromethION Flow Cell loading with the demo pack, I just wanted to take a quick note on how to use the syringe as a pipette in our particular case. As you can see, the syringe has a plunger and it has a grading scale from 0.1 to 1 milliliter in 100 microliter increments. To be able to correctly measure out a certain amount of liquid using the syringe, what we will need to do is we will need to press down the plunger all the way to the very bottom, and then as we're aspirating the liquid, we will need to focus on the very widest part of the plunger that almost looks like a mark, the one that goes and surrounds all the walls. If I flip it over, you will see it in a little bit more detail here. So this part of the plunger is the one that's used for measurement. There will be some dead volume, some air inside of the syringe. This is absolutely fine. We don't need to worry about this. It is just dead volume inside of the syringe. Now I will push the plunger all the way to the bottom so there is no unnecessary air in the syringe, and we can bring our flow cell back. Now, as a very first step, if you remember correctly, the first step to do on the flow cell loading side, we need to open up the priming port cover to expose the priming port, and we can already see that the priming port itself has a gray colored channel next to it. This gray colored channel suggests that we have a little bit of air inside of the priming port. Now, we don't want any air inside of the flow cells because they can damage the arrays, as well as air and air bubbles do not pass current very well, so they can just block the channels from passing the electrical current. So as the very first step, we need to remove that air. To do that, I will make sure I press the plunger of the syringe to the very bottom, and we insert the syringe into the flow cell, and very carefully, using our thumbs, start aspirating liquid up. And as we can see, we have a little bit of storage buffer enter the pipette tip of the syringe, and this suggests we have successfully removed the air bubble and actually reached the storage buffer that was inside. At this stage, we're ready to do the flow cell priming. Now, I will use the said tissue to expel any excess liquid out of the syringe and proceed to take our priming mix, which is the flush buffer here, to do the priming steps. The priming of PromethION Flow Cell is done in two steps of 500 microliters. So I will open my tube and aspirate 500 microliters or half a milliliter into the syringe. Before we do that, let's make sure that our plunger is at the very bottom of the syringe. Insert it into the tube vertically, and very carefully and gradually let the widest part of the plunger reach 0.5 milliliter mark. Once we reach that, we can once again take out the syringe out of the tube. Check that there is no air in the very bottom of the pipette tip, and once that is done, we can confidently insert the tip into the flow cell and carefully press down onto the plunger until the level of liquid reaches somewhere in the middle of the pipette tip. As always, it is okay to leave a little bit of liquid in the tip, so we can disconnect it. We can disconnect the tip, and as you can see here, I'll show it a bit more. There is still some liquid, some priming buffer left inside. I will let everyone finish with this part. And we have done the first priming step. Now, normally there would be a 5-minute wait step, a 5-minute incubation step located here, but because it is a demo flow cell and we're doing it only for demonstration purposes and for learning purposes, we can snap our fingers and use our time bending abilities to make those 5 minutes pass faster. Now that those 5 minutes have passed, we can proceed with the second priming step. So what I will do is I will take my syringe, insert it once again into the tube with liquid. I will make sure, of course, that the plunger is pressed down all the way to the bottom. So we insert it down and carefully aspirate the liquid up until the point where the widest part of the plunger reaches 0.5 mark on the syringe. I'll take out the syringe, check for any air in the pipette tip. I don't see any air in mine, so I know I can proceed with the second step of loading. So I insert my syringe in and carefully dispense the liquid using the plunger until about halfway down the pipette tip. Once again, I always leave a little bit of liquid in, so we can see we have liquid still present in the pipette tip. Hopefully everyone has done this part again. I will wait for everyone to finish in case you are still doing that. And at this stage, our flow cell is successfully primed. Now, as the very final bit, we need to add the DNA sample and library into the flow cell. As our DNA library, today we will be using library loading beads. As you can see, if you look at the tube, it may not be visible on the camera very well, but on the ones that you have, it will be visible. There is a clear phase separation. This is because the library loading beads fall down to the bottom of the tube very, very quickly. So, and this will also happen with your real samples when you mix your DNA library with sequencing buffer and library loading beads. So we recommend to mix and make sure that the beads are resuspended. And today we can do that by gently flicking the tube or inverting it a few times, like so. Once it is mixed, you can visually check the tube that the liquid is now resuspended and homogeneous. There is no phase separation, and we can load our library onto the flow cell. So once again, I will press the plunger of the syringe to the very bottom to make sure there is nothing else in there. Open the tube with my library beads. Insert the pipette tip vertically into the tube, and this time aspirate 200 microliters, so until 0.2 mark in the syringe, to take 200 microliters of material of our library into the syringe. Once again, let's check that we don't have any air inside of the tip. Insert the tip into the flow cell and carefully press down onto the plunger to expel everything into the flow cell. Now I will once again stop just a little bit before and take the syringe out. Once again, this way, by not loading the liquid fully, we can make sure we're not introducing any air into the flow cell. At this point, I'll close my tube, and we have loaded the library. Congratulations. Now we will close the valve, and at this point, we are finished. We have loaded our first PromethION flow cell using the demo kit. Now, if this was a real flow cell, we would be able to put it onto the device, but because this is a demo flow cell, it cannot be put onto the device, so I will just take the pouch that everything came in with, and I will dispose of flow cell, the reagent tubes, and the syringe, as well as the tissue, if you have it, back into the pouch. So I'll just pop it in here and seal the pouch to make sure I can dispose of it in accordance with local regulations. Thank you very much. Congratulations on loading your first PromethION flow cell. Now, to make sure that everything is clear and you are able to do this and repeat it many times with the real PromethION flow cells, let's go over the few last key points of PromethION flow cell loading. So, if you're loading a flow cell inside of the device, we would recommend to make sure that the flow cell is positioned correctly. If you're loading the flow cell on a table, please make sure it is positioned horizontally and on a level surface. As a very first step, open the priming port cover and remove a small amount of air, a small air bubble from the priming port to make sure this bubble doesn't get onto the flow cell. Whenever loading any liquids into the flow cell, we recommend to not expel all the liquid out of the pipette and leave a small amount of buffer or library inside of the tip to avoid, again, any air going into the flow cell. Once all the loading is done, ensure the loading port is sealed and closed so nothing evaporates over the 72-hour sequencing run and you're not changing, and the osmotics of the flow cell are not changing. And at this point, the flow cell is loaded and ready to sequence. Typically, before starting sequencing, we recommend to wait for 10 minutes for the library to stay on the flow cell before putting it into the device and loading. Hope that was easy and clear. And now, in the next masterclass, my colleagues will introduce you to how to use MinION software to set up a sequencing run. Because I do have a flow cell already inserted here in the P2i device, what I will do is I will just quickly wake up the screen and click Start the sequencing run. But for more details, please stay tuned for the next masterclass where more details will be provided on how to set up the sequencing run in MinION and how to use MinION software. Thank you very much for your attention. If you'd like to learn more about PromethION product range, please check out the QR code on your screens right now or visit nanoporetech.com/PromethION. Thank you very much and all the best luck sequencing.